afternoon, everyone. So what we will cover today will focus on a quick review of genes and evolution of genes, as that is important as we use sequence data to assess relatedness of organisms. The bulk of it will then focus on data analysis approaches, including how we use these sequencing data to build trees and how we interpret trees to support epidemiology of infectious diseases. This is a slide you've probably seen multiple times to remind ourselves of what we generally talk about and what type of information we generally use when we try to decide whether two or more organisms are close related, the same, or very different. When we look at genome sequences, we typically look for single nucleotide polymorphisms, or SNPs, as shown at the top of this slide, where we have two sequences and they differ by a C in the top organism versus a G in the bottom organism. Now, what we also often see is that there are other types of changes. In the bottom, we see what's called an indel. It's an insertion or a deletion, where rather than a nucleotide changing from one to another, we simply have one missing nucleotide. Evolutionarily, we can't necessarily know whether that means a deletion in the bottom or an insertion in the top sequence, so we simply call them indels. But as we try to use whole genome sequence data, so long stretches of these nucleotides, ranging from a few thousand for total viral genomes to millions and beyond for bacterial and fungal genomes, we really need to look a little bit deeper in terms of what the different mechanisms are, how organisms can become different. The ones I showed you before, the single nucleotide polymorphism, can be simplistically thought of as having a parent organism, let's say a bacterium shown on top, that then divides into two bacteria, where one of them has a change, so a nucleotide changes from whatever it was originally to an A, as shown in black here, in red here, and that then creates one SNP by one genetic event, one mutation. Now, what we can also see is insertions and deletions, as shown before. Now, these insertion and deletions typically occur when we have stretches of nucleotides that are similar to each other. So here the parent sequence shown on top has a repeat of TAG, TAG, TAG. So it's fairly easy for an organism to not replicate this sequence accurately, so that rather than 3 TAGs, we have 2 TAGs as shown on the bottom left. Now that can be seen as one genetic event. We just dropped 3 TAGs during replication, but it also could look like, if we don't know that, as 3 differences. So we can start to see that one event may create more than a single SNP and can, in this case, as shown in this case, create 3 differences. Now, there are other mechanisms that can create differences. One of them is an inversion, where we take a piece of DNA, shown in italics, in top sequence, and just flip it over by 180 degrees. That then creates a set of differences, and if we align them, as in this case, we can see we have 2 SNP differences. Some of the nucleotides still match simply by chance being the same, but we can have this one single event where we flip over a sequence, creating actually 2 SNPs. And finally, maybe the most important one as we look at epidemiological information is horizontal gene transfer. Horizontal gene transfer means that we move genetic material from one organism to another distinct organism. Textbook example that is easy to envision would be plasmids, for example, where we can have an organism that is sensitive to antibiotics, but it can acquire a plasmid, which can be thousands, tens of thousands of nucleotides, and with that get a lot of new genetic information, plus resistance to antibiotics. The one event, horizontal gene transfer, that becomes more challenging as we interpret DNA sequence data is horizontal gene transfer of homologous gene sequences. What that means is, when we look on the left side, we have one bacterium. It has a gene with a certain sequence, shown in black. Now we have a similar bacterium that lives in the same environment that has the same gene, but with slightly different sequences, and the differences are shown in red here. Now that sequence can move from the one bacterium shown on the right into the one on the left, incorporate into the chromosome, and replace the sequence that is there in one fell swoop. So with one single event, we can introduce a considerable number of differences. In this case, when we do an alignment between the original sequence and the one that happened over one generation, we find 3 SNPs that occurred very, very quickly. So these 2 organisms, in a matter of potentially a few minutes, can become very, very different looking if we do genetic analysis and whole genome sequencing. And really a lot of the analytical methods we use are designed and have to deal with these sort of horizontal gene transfer events to make sure they don't confuse us as we try to derive epidemiological relationships from whole genome sequence data or as supported by whole genome sequence data. How does this sort of moving of DNA pieces work? In addition to plasmid transfer by a mechanism that is called conjugation, the 2 key mechanisms are transformation and transduction. Transformation simplistically will be if a bacterial cell, like the one shown in black, dies, the DNA fragments, and another bacterium that is still alive, as shown in red, just takes up a small piece of DNA and then incorporates it in its chromosome, replacing whatever genetic material is there with a different piece of DNA. The other mechanisms occurs via phages. So phages are bacteria that can infect, or viruses that can infect bacteria. And when they infect bacteria, they can transfer pieces of DNA, anywhere from fairly small pieces to longer pieces, or even the whole genome and insert it into the genome of the bacterium. So 2 distinct mechanisms of how this DNA transfer happens. So it's not just a theoretical construct, but we understand the biology of how big pieces of DNA can be moved around between different bacteria very, very quickly, and therefore creating potentially considerably different sequences very fast. Now, this is a practical example that actually happened to us. In this case, we investigated retrospectively a human listeriosis outbreak that occurred in 2000, where we had 29 human cases. When we analyzed selected these isolates, one of these human isolates showed a single SNP difference, so a difference in 1 nucleotide over 3 million nucleotides, to food and human isolates from a single case that was linked to a processing facility in 1988. So 12 years difference, we find an isolate that matches this outbreak. Epidemiology supported that this facility was the source of the outbreak, suggesting that that Listeria survived in this facility over 12 years, caused an outbreak, and combination of epidemiology and whole genome sequencing really allowed us to draw this conclusion. Now, if we would have had approached this analysis differently, we could have seen that there were 3,000 SNPs because there was a massive, large recombination event that with the wrong analytical approach, with the wrong approach to analyze these genome sequences, we would have decided that these sequences are very, very different. So these ideas of recombinations obfuscating relationships by introducing considerable amounts of different DNA is not just theoretical, but it is something that has shown to cause problems in epidemiological investigations. And for those of us used to PFGE, very often when we see and when we talk about the 3-band rule, if we have organisms that differ by 3 bands, those are typically and very often also caused by recombination events. So how do we apply this information as we try to assess the relatedness of organisms? These differences in sequences that we can see and that can occur by different mechanisms can be used to assess the relatedness of organisms and the likelihood that 2 bacteria or 2 other organisms, 2 viruses, 2 fungi share a recent common ancestor. And that's what we typically do when we analyze these data. So we ask questions like, do the Mycobacterium tuberculosis isolates from patient A and patient B share a recent common ancestor, so that we can look at both genetic data, whole genome sequence data, and epidemiological data. The key here is that the definition of recent common ancestors becomes important. We can look at recent in 2 different ways conceptually. One of them is, and most of us initially will think about it, as recent in years. Do they share a common ancestor that occurred within the last 2 or 3 years? The challenging part in the analysis we do here is that it's much easier to find recent in terms of generation times. how many generations back did these two Mycobacterium tuberculosis isolates come from the same bacterial cell. When does it become very important? We can take the same organism in different environments, and there can be a huge difference between recent in years and recent in generation times. Take a salmonella that lives in a dry processing plant, such as a plant that makes cereal. That salmonella may stay dormant, rarely if ever grow, and might only multiply 100, 200 times a year. On the other hand, if I have the same salmonella in a chicken flock, where I have active infection, it could theoretically multiply every 30 minutes and multiply 7,500 times a year. So depending on the environment, the amount of genetic change that is expected over a year can differ considerably. The only way to get that information is if we have epidemiological and source data that we can look at and take into account when we interpret some of the whole genome sequence data. So the bottom line here is that assessing relationships of microbial isolates typically requires more information than just sequence data. I gave you one example just a few seconds ago. Another one is, of course, is that we need epidemiological data to identify two relationships. We can have bacteria that are basically appear to be the same, and they share the recent common ancestor, maybe in the last 2 years, but that does not necessarily mean there's an epidemiological relationship, because bacteria can disperse over long distances very quickly. Simply think of things such as migrating birds that carry bacteria. So there are mechanisms that allow for rapid dispersal and therefore allow us to find the same bacterial strains as determined by whole genome sequencing in different places. So with that, we move into data analysis approaches. How do we take whole genome sequence data and large-scale genetic data to assess relationships between different organisms? Conceptually, there are a number of different methods and two key methods that will be used in terms of analyzing data that are handled by epidemiologists today. The two key methods are high-quality SNP analysis and whole genome MLST analysis. Sometimes we might see other analyses that are called k-mer-based analyses, but these are really not typically used for epidemiological data analysis and typically only provide a rough grouping of isolates. So we will focus on high-quality SNP and whole genome MLST analysis. Importantly, for both of these analyses, we utilize the same raw data. When we perform and when the lab performs the whole genome sequence data, the same set of data can be used to do high-quality SNP or whole genome MLST analysis. It can be used to do high-quality SNP analysis today and whole genome MLST in a week or a year, and vice versa. Also importantly, both high-quality SNP analysis and whole genome MLST analysis provide data on the number of differences between isolates that can then be used to build so-called trees, which we will show you, that can be used to more visually assess the relationships between isolates. So the first part I'm going to cover is high-quality SNP analysis to identify differences between isolates, and that could be bacterial isolates, fungal isolates, viral isolates. So what makes a SNP, single nucleotide polymorphisms, high quality? The idea is basically to apply certain filters that take out nucleotides where we're not as sure as we want to be that these are correct. So we take all the differences, all the potential nucleotide differences, and filter out and apply quality filter to get the sequence reads that are ready for analysis. And we have more details on that into some of the modules that are available, as Genevieve mentioned earlier today, and I will not go into depth into these. So next-gen sequencing of a genome can generate 100,000 or more reads per genome. And so in this case, the example would be a bacterial genome that could be anywhere, typically would be anywhere from 3 to 6 million nucleotide long. Any single location, so any gene, any nucleotide on the genome, can be covered by more or less sequence reads. What we see here in yellow we see one coverage on the left side that is called coverage 40x. That means we have 40 sequence data, 40 individual data sets to look at one nucleotide and to determine whether that nucleotide is an A, C, T, or G. On the other hand, we see on the right-hand side, also covered in yellow, an area that has much sparser coverage. So there we might only have 5 sequence data to determine whether a nucleotide is an A, C, T, or G. So one very simple way to look at quality is to say, we're only going to analyze data where we have multiple SNPs and a certain number, multiple of coverage, a certain number of coverage. We could set a coverage cutoff, for example, at 25 folds or 25x. So that is one way to filter out lower quality SNPs. An additional way, in addition to coverage, is to use space frequencies to define high quality SNPs. If we have 20x coverage, we can apply a criterion saying we're only going to call something a certain nucleotide if 19 out of the 20 repeats agree. So we see this on the left. On the right side, on the bottom, we see a reference sequence. On the top, we see multiple coverage of the same site. And one of them you can see we have a lot of sequences. Once it's an A, once it's a T, and all the other 8 times it's a C. So if we say we have to have 95% consistency, this might not be a high quality SNP. If we have a cutoff that is slightly lower, let's say 80%, we could call it a high quality SNP. So we set off cutoffs right there to define a high quality SNP. On the right hand, you see one that is consistent, but it has a lower coverage. So coverage and base frequency go together. This is not something that typically on the epidemiology end of things people have to deal with, but it is important to realize that these analysis in terms of defining high quality data do occur before the final analysis are done. The other questions, and that gets back into some of the discussion we had about recombination, is that not all SNP pipelines are equally, and where specifically called SNPs affect the total SNP count. Well, the second bullet specifically outlines this problem, and this is where we say that SNPs that are relevant and that are used for phylogenetic analysis are those that are vertically transmitted, not the ones that are moved around horizontally through things such as phages or genes that are affected by horizontal gene transfer. So in a high-quality SNP pipeline, when you typically want to have sufficient biological and genetic understanding of the organism to be able to mask certain mobile elements. So you mask an element, say, we're not going to call SNPs in this specific area because we know it's a mobile element. And in the example that I gave earlier today, we would mask the specific area in the genome of the Listeria isolates where we know it represented a phage, and that took then out 2,999 or around 3,000 SNPs because they were in one of these mobile elements and therefore would have confused the analysis and called two isolates very different, when in reality the difference happened very quickly through one of these mobile elements that can move around very easily. So that's one of the other key parts of a high-quality SNP is that we remove certain elements and don't use them to identify SNPs because we know that it can change quickly. How do you report SNP data? And this is some examples that we got from some of our discussions with our colleagues in Minnesota. Sometimes, despite all the complexity of the analysis, it's actually fairly simple to communicate results. For example, if you have a high-quality SNP pipeline, a simple email could go out saying to a set of epidemiologists or whoever we report this, saying, we've just recently had 2 isolates that are 0 SNPs from each other, basically identical. They represent a putative new cluster, and here are the isolate names. Or we can say, we got a new cluster, we have 2 isolates that are 2 SNPs from each other, and report that. In this case, the lab typically will make a decision that occurred over iterative discussions with epidemiologists and other data to say, with a certain SNP cutoff, at for example, for Listeria, might be 5 to 7 SNPs, that anything that is more similar than that is considered a cluster that may warrant an epidemiological investigation. Another way to look at SNP differences is by these massive similarity matrices as shown here. Now, they might be used by some epidemiologists and labs as they communicated with each other and might not be used by others, but I think they illustrate a relevant and important concept. Here, color coding implies the level of differences, and I'm going to pull out one set on the red side and show you what that specifically looks like. Here would be one of these SNP matrices. We can compare two isolates. We have the names, and we can say these two isolates differ by 826 SNPs. So these isolates are very, very different. So slightly more complex way to communicate, rather than saying here are two isolates that are the same, but it allows epidemiologists and end users to look at these data and identify the SNPs between any types of pairs of isolates that one is interested in. Now, if we look on the other end, where we have very similar isolates, so the white side of that similarity matrix, we can see isolates here, and we can see that they all differ by very few differences, from 0 to 4 SNPs. And again, we can compare two isolates and say this isolate and this isolate are basically or identical with regard to number of SNPs, 0 SNP differences. So in this case, there's more opportunity for the end user to really look at all data by getting these similarity matrices. The more data we have, the more isolates we have, the more complex it obviously gets to look at those. So if we summarize high-quality SNP analysis, the advantages of these analyses are that they provide phylogenetically informative information. So they are consistent with the evolution of these organisms that we look at. It requires, as a disadvantage, it requires a closely referenced genome, so a genome that we sequence very well, where we have a considerable amount of information to provide the high-quality SNP analysis. If we do not have a reference genome that is similar to the organisms we analyze, these analyses can become challenging and difficult. So if I, for example, would want to analyze a genome for Salmonella Oranienburg, and the only reference I have is Typhimurium or something like that, then it might not work as well. It is high-quality SNP analysis is very good for situations where we don't have a whole genome MLST database, but the database is in the process of being developed, and in some cases might provide the best resolution for strain comparisons. SNP position can be identified on a genome, so we can figure out which gene is affected by a difference. That can be valuable as we identify differences. If you have a food and a human isolate, for example, it differs by a number of SNPs, and these are SNPs in a surface molecule that are target for the immune response. We can logically deduce that some changes in this domain in this gene might happen during passage through a human. These analyses, on the other flip side, though, are typically more time-consuming. We're not talking about days here, but they're more time-consuming than quick whole genome MLST analysis, and can require a considerable amount of computing power, particularly as we compare more and more strains. Maybe one of the biggest challenges is that the interpretation of data depends on the number of genomes added. If you build a tree and you add 10, 15 more sequences, the tree may not look exactly the same. It may not be easy to simply assign a number to a subtype and have these numbers meaningful so that we know if two isolates differ by a few SNPs or by thousands of SNPs. So very sensitive, very good from an evolutionary approach. In a high throughput fashion where you want to have a lot of different people use it, may not be the most feasible method. And this is where the next method comes in that we're going to talk about. That's whole genome MLST analysis. As compared to high-quality SNP, where we look at the whole genome, come back to my favorite organism, Listeria monocytogenes, we compare 3 million ACTs and Gs. We now split up those 3 million ACTs and Gs in genes, which an average gene being about 1,000 ACTGs long, and do the analysis gene by gene. So rather than one big analysis of 3 million, we analyze 3,000 times 1,000 nucleotide sequences. So the analysis is split up into smaller pieces. That's the essence of whole genome MLST analysis. This relates back to traditional MLST multilocus sequence typing. This was a method that was developed before the wide availability of next-gen sequencing, or before the availability of next-gen sequencing, and was based on Sanger's sequencing, the traditional sequencing methods. In this case, people would classically sequence 6 to 7, and sometimes more, housekeeping genes. So housekeeping genes being genes that fulfill key functions, and they are found in all bacteria of a given species or target group. So we pick 6 genes that are found in all Salmonella, we sequence them, and use that to determine similar, same, or different. The idea was that sequencing multiple genes that are found in different places of the bacterial chromosome, so they're not all next to each other, allows for improved discrimination over sequencing one gene, and also potentially reduces the risk of one gene being different simply by this gene being horizontally gene transferred, or something weird happened to that one gene. So it reduces the risk of error. In general, the experience with this approach has been that it is a relevant and a very good method to understand evolutionary relationships, but typically did not provide the appropriate discriminatory power to help with outbreak detection, for example, with Salmonella or Listeria. But the general scheme has been well test-run. The schemes that we internationally agreed on available, they're publicly available databases that make it fairly easy for people to analyze these data. And we can build on that past experience and history to now build out these MLST to not just look at 6 to 12 genes, but look at large numbers of genes that we derive from whole genome sequence data. Typically, this combination of 6 to 12 genes is used to name a unique sequence type, which is a unique combination of genes. The 6 genes are the same, the isolates are given the same sequence type, such as in this case, sequence type 1. How do we apply this to whole genome MLST? We build a database where we catalog all the genes in the organism of interest, that would then be called whole genome MLST, or all the genes that are found in a core set or the core genome MLST. Whole genome MLST is the one that is more commonly used. Each unique gene we often refer to as a locus. A locus may be the complete gene. It could be thousands of nucleotides. Sometimes might be a piece of a gene or some other part of the genetic sequence. Any changes, we talk about different mechanisms of changes. SNPs, insertions, deletions provide a new allele call. So we see this on the bottom when we talk about locus 1. We have the first allele. We sequence the second isolate, and it's different. By 2 SNPs, we call it allele number 2. We have a third isolate on the right side. It's different from the other 2. By deletion, it's called allele 3. The important thing is that the numbers are consecutive on occurrence when we see it, not by difference. So allele 1 and 2 could be different by 20 SNPs, while allele 1 and 500 might differ by only a single SNP. So don't look at numerical proximity and numerical closeness as a measure of actual relatedness of the isolates. When we do this as a large scale on whole genome MLST, we have a set of loci. It can be of different length, and this is hypothetical. We have different isolates. If isolate A, B, and C have the same sequence of the first locus, it's all allelic type 1. At locus number 2, isolate A and B are the same. They're both allelic type 8. Isolate C is different. It's allelic type 12. We then combine all that information onto a whole genome MLST type. In this case, isolate A and B would have the same combination of 2005 numbers, and therefore would be given the same whole genome MLST type. The way this looks when we then move it to a larger scale and types of databases that are used, this is what we see. We have a tree. We have the different loci. In this case, they're from Listeria monocytogenes, therefore LMO. And we can see on the LMO1 locus, all these isolates are the same. They're allele 11. LMO2, they're all the same. But then as we go further down, we see differences. And this list obviously goes on and on, not just to LMO17 on the right side, but to LMO more than 2,000 probably. And so then we can use these data to also build these trees that are shown on the left side. In order to create naming patterns that make it easier to compare allelic types, there have been some naming schemes proposed, including one that will be used, it's my understanding at least, for some of the foodborne pathogens where this approach will be applied, is that we name these alleles with multiple numbers, the so-called zip code system. If two isolates have exactly the same numbers, they're exactly identical. If they're different in their last number, they're different by 7 alleles. If they have the same 3 numbers and they're different in the last 2, they're different anywhere from 8 to 15, etc. So they're the numbers that are designed in a way that they give you some information on similarity and difference between two isolates. How does that work? Here's an example. You have 2 isolates. We did whole genome sequencing. We ran whole genome MLST. Isolate from patient 1 and 2 are the same. They have exactly the same zip code, exactly the same combination of 6 numbers. We have add a third isolate from a third patient. Now that patient isolate is slightly different. It differs by the last number. That means patient 3 differs by anywhere from 1 to 7 alleles from patient 1 and 2. We add another one to it. Now here the last 2 numbers different from patients 1 and 2. So that means the isolate from patient 4 differs by anywhere from 8 to 15 alleles from patient 1 and 2. So you can see how this numbering system and this zip code system makes it very easily to just look at 8 numbers to help you assess the similarity of isolates from different patients and different sources. So here how we report these data. Again, we could stick with a very simple scheme of reporting data that just tells people that this is the slide I showed. So we can simply report results the same way I showed you before, where we just say here's a cluster, and these 2 isolates differ by 0 to 2, 0 alleles, or for the cluster on the bottom, these isolates differ by 2 alleles. Let's see, getting used to this other computer, or we can report the actual what we call the zip code data. So we can provide data on a set of patients, saying here's patient 1 through 5. And from this, you can see that patient 1 and 2 have basically, or within 7 alleles from each other, and then the end users can actually interpret these patterns of zip codes to see which ones they want to investigate. In this case, obviously, patient 1 and 2 would be very similar to each other with regard to the isolates, while the isolate from patient 4 and patient 5 are very different from the first 2 patients and would not be expected to be part of the same cluster. If we summarize MLST analysis, one of the major advantages that it tends to be faster than analyzing SNP differences. For whole genome sequence data, these allele calls, so where we analyze data sequence by sequence, can be performed on the actual short reads that come straight off the whole genome sequencing equipment, or we can perform them on assembled genomes. So it provides another option of how this data analysis can be performed more quickly. Typically, the system that it looks like we will have going forward is that these allele calls will be performed both assembly-free very, very quickly, and then on assembled genomes, which probably takes another couple of hours. And if these 2 data do not 100% agree, then for that specific allele, for that one gene, there's no allele call made, and the analysis is continued without the call for this one gene or this one locus out of 2,000-3,000 loci. So here's a summary of the advantages and and caveats of whole genome MLST. It also provides phylogenetically informative information. We can pull out very quickly genes that allow us to determine serotypes and antimicrobial resistance. It has an effective way to neutralize the effects of horizontal gene transfer. If two genes or if one locus is different by a lot of nucleotides between two isolates, it's really only counted once because we don't count the differences in SNPs, we just count the difference in alleles. And maybe the biggest advantage is that it allows for stable nomenclature, the zip code system that I showed you, that really will make it very easy to communicate results between different labs and from labs to end users, such as epidemiologists. I'm going to skip over the rest of them to make up some of the time we lost. If we then compare high-quality SNP versus MLST analysis, both analyses, as I said before, can be conducted using the same raw data. So we start with the short read sequencing data and then analyze them further. Very importantly, for public health purposes, in most cases, we find that both of them correlate very well. The outermost branches of phylogenetic trees, the ones which show us which isolates are really very similar, are typically identical. But for some cases, and it could be for some types of organisms, etc., MLST may work better. For other ones, SNP may work better. And that is going to become more clear as we do more large-scale analysis on more different types of organisms. But bottom line is basically both of them will work, typically give you very similar or the same results, but in some cases, it might be helpful to run both analyses. Now, the last part we're going to go is how do we analyze these data in terms of building trees based on the whole genome sequence data. So both wgMLST and whole high-quality SNP provide us the basic data that we can use to infer the relatedness of isolates. High-quality SNPs provides us some additional information that allows us to really fine-tune how we determine how similar isolates are. And then at the bottom here, I'm giving you one example of that. So we can see we have 4 nucleotides, A, C, T, and G, as shown below. What you can see from the structures below is that they actually look different. And what you can in particular see is that thymine and cytosine, so T and C, have a similar structure, and A and G have a similar structure. So from this, for example, you can see that a change from a T to a C is more likely to occur, the structures are similar, than a change from a C to an A. Those sort of subtleties in terms of the likelihood of changes occurring can be and are typically used in the high-quality SNP analysis. So here are the basics of a tree. I gave you an alignment of 4 sequences, 1 to 4. I highlighted the difference in red, and we can have a computer program build a tree. Now, if you look at the right side and you look at those trees, initially they might look different. In reality, these are the same trees. What happens with trees is that you can actually rotate trees at each node. So you can see a node is basically where the tree splits, and every one of them you can rotate. This is important because it's a note of caution that if you look at relatedness, you cannot just look up and down and say, well, 1 and 2 are next to each other, 3 is next to 2, and 3 is next to 4. So 3 and 4 got to be pretty similar. Because if I simply rotate the tree as shown on the bottom, now 4 and 3 are very separate from each other. But it is the same tree, it is simply rotated. So when we analyze a tree, resist the temptation to just look up, down for similarities. You really need to look inside the tree to get good and appropriate comparisons and identification of which isolates are similar to each other. So here's an example tree that we're going to discuss more over the next few slides. So this gives us the relatedness of 4 isolates, A through D. It has a tree, but it also shows us the number of differences. So for example, isolate B here is different by 6 from the common ancestor at the node where the 1 is shown. So this is your base tree that simply gives you similarities. And the relative relationships of these isolates. So you can see that isolate B and C share a common ancestor. They come from the same node. But you can also really see that isolate A and C, if you look at the number of differences, are actually more similar to each other. So B and C, same ancestor, so have a recent common ancestor, but B accumulated a number of changes that could, for example, happening by isolate B growing very, very rapidly in a certain environment and therefore accumulating more changes. If you talk about trees, here's how we refer to them. I already used the term node, so that would in this case be the most common ancestors for isolate B and C. Leaf or taxa is basically the isolate that we have, and then a group of sequences that have a common node referred to as clades. Importantly, the trees can be displayed in a number of different ways that I've shown here. The ones around the circle really are all the same trees. They're just twisted in different ways and therefore might look very different, but in reality, they're the same trees. In the middle, we see, in the colored, we see a minimal spanning tree, which is another tree that can be used and typically is used with MLST and whole genome MLST data. And we'll show that later again to explain it. But the important thing is when you get trees, when you look at trees, they may look very different, but the basic rules of interpretation apply to all of them. So if you look back at the tree that we started out with, so this tree shows us that isolates A and C, as I mentioned before, are more similar to each other than B and C, but isolates B and C are more related to each other to share a more recent common ancestor. Again, that might sound very counterintuitive. How can they share a recent common ancestor and be more different? But we don't have all individuals in the population, and B just could have evolved very quickly and could have been removed a number of generations from that common ancestor. We then can map epidemiological information on these trees. So here we now put the sources of the isolates on the tree, and we can see we have a stool isolate, spinach isolates, with the stool and spinach isolate sharing a common ancestor. But then we have a kale isolate that is very close to the stool isolate in terms of similarity. And we're talking about the stool isolate in green on the top. So what this shows is that with these data, with a tree like this, we cannot really identify that whether spinach or kale will be the source of the salmonella in the stool. It could actually be none of these two isolates. It is epidemiological data that are required to then make sense out of these patterns and out of these evolutionary relationships in terms of a cause and effect relationship, which food could have caused the human case that then led to this stool isolate. Here's the idea of the minimum spanning trees, which is used for whole genome MLST data. And so here, the length of the branches between the different isolates that are shown as blue boxes indicates the number of allelic differences. It's very small, but the number is typically there. And we can see a number of isolates on top that are all very similar to each other. And again, through a combination of that tree and epidemiology, we can show that these isolates are all linked to one outbreak. Here's then the trees that one would more typically see as we start to accumulate more and more data and communicate results to end users, such as epidemiologists or others. You see a number of outbreaks that started as clusters, but then became outbreaks through epidemiology and marked in different colors. So if you look at the one on top in purple, you have 4 isolates that are very similar to each other, 0 to 2 SNP differences. Another set as we go down, so the number of SNP differences are shown in these, and the number of isolates that group together as a clade with very few SNP differences and as having a common ancestor. But you can also see, if we look at the second outbreak from the top, we have 4 isolates that group very closely together, but one of them is a sporadic isolate that epidemiology did not link necessary to the other isolates in this outbreak. So epidemiology, again, is essential to make sense out of these data in the communications between epidemiologists and the lab and environmental health or environmental investigators, if we investigate environmental sources, are very important to help us the interpretation of these data. So the take-home messages from this is really that molecular epidemiology, and particularly application of these whole genome sequencing and next-generation sequencing tools that can be used to sequence things other than bacterial genomes, require collaborations between epidemiologists and the lab, and only both data sets, epidemiology data sets and evolutionary relationships, can be used to resolve outbreaks. One alone, while epidemiology alone can detect some outbreaks and always has, lab data alone can rarely, in my mind, if ever, be used to really define an outbreak source or define even an outbreak. The important thing to keep in mind is that microbial isolates can accumulate genetic differences through a variety of different mechanisms. Some of them change them slowly over time, such as single nucleotide polymorphisms. Other ones can allow for considerable changes very quickly due to horizontal transfer from one cell to another cell of a large chunk of DNA. The approaches that data analysis use to deal with these evolutionary mechanisms can play an important role as we interpret these data, and the analysis approaches that I showed you, high-quality SNP and whole genome MLST, typically address, and both of them truly address these issues and account for horizontal gene transfer. Not necessarily in exactly the same way, but they account for them and therefore make sure we have reliable data in virtually all cases. The one I didn't mention, ksNP, would not account for these horizontal gene transfer data, which is one reason why it is not used very much. Now, the important thing is that as we apply these tools to other organisms that we may not have worked with, organisms with different their lifestyle, and I explained to you that the number of generation times that an organism duplicates can be very important in terms of the number of differences accumulated. We may have to reevaluate how we deal with these data, how we interpret these data, and maybe even what analysis we approach, what analysis we use. The key is that we need to know and understand the epidemiology. We need to use epidemiology along with whole genome sequence data, and we need to understand the biology of the organisms we deal with to allow us to accurately interpret the data and come to meaningful outcomes, which is rapid detection of outbreaks and outbreak sources. With that, I want to thank our colleagues at the CDC for providing slides and providing inputs on the slides, and the Integrated Food Safety Centers of Excellence that have brought these slides and this presentation to you. With that, I'm going to open it up for questions, and which you can type into the interface under the chat or the Q&A session. So one question is that if one would have some isolates that are very, very clearly related by epidemiology, very strong epidemiology, let's say it could be 2 human isolates, could be a human or food isolate, and they show a large number of SNP differences, let's say more than 40 SNPs. Could this be because of recombination? Would there be other reasons for that, and how would one handle this? So it obviously depends on the actual data analysis used if you have a situation like this. But a situation like this could definitely be a horizontal gene transfer event, an unusual genetic event, as I might call it, that was not picked up and accounted for correctly in the analysis. That's rarely to occur, but I think as we analyze large data sets, that could happen. So I think it's another example where clear communication between epidemiologists and end users in the lab comes in. Go back saying, you know, we have very strong evidence of epidemiology relationship. You tell us there are 50 SNP differences. Can we talk about this? What could have happened? This is also where observations become very important. If you envision a situation where this might be from a patient, one isolate is from a patient that has been treated with 3 or 4 different sets of antibiotics, because of various reasons, certain conditions that may affect the number of mutations that one could see, then that would be important information that wouldn't be needed to assess that. Of course, there are other possibilities. One of them is that there's strong epidemiological relationships, but one of the sources had multiple different subtypes. Maybe one in the food had 2 salmonella in it. We picked one and we picked the wrong one. Not that there's a wrong one. We picked one that was not related. If we would characterize more isolates, we might have another one that actually matches very, very closely or exactly between the 2 sources. So a number of different possibilities. Horizontal gene transfer definitely could be one, but the key is this is where these communications become very important. We had one question was for the wgMLST zip code. If 2 zip codes are only different by 1 number, why does it translate into 1 to 7? Okay, if I said that as misinterpreted, if 2 zip codes are exactly the same, that means the isolates will differ by 0 to 7. They're either exactly the same or have very few differences. If they differ by 2 numbers, it's 8 to 15. The differences actually will depend on the isolate. We cannot provide an exact number of differences because then we would have to have a too long a zip code. So we bin the differences into different groups. Very few differences. In some cases, for some organisms, it might be 0 to 7. More differences, but still close related, 8 to 15, etc. This will depend on the organism, and the cutoff will be dependent on historical data, the evolution of the organism. So, for example, for Listeria, where I showed you the example, large sets of analysis on thousands of isolates have been conducted, and they basically consistently point out that isolates that differ by up to 5 to 7 SNP differences are very often likely have a recent common ancestor and might be part of the same outbreak. So these evolutionary data from outbreak investigations drive what these different bins are that we put differences in there. But zip codes provide a range of differences. Another question is it possible that an ancestral root defined by generation time could have come after another isolate when defining time in terms of years? Trying to make sure I get this question right. The ancestral roots definitely are defined by, initially measured by, it can be measured by different ways. If we have enough isolates, we can try to design it so it represents years, but the assumption, if the length is shown in years rather than number of SNP differences, is that we have a consistent generation time. If we mix and match isolates where some of them might be very rapidly grown in a very environment that grow very rapid, other ones are in an environment where they grow very little, then these scales may not be very meaningful. So as we try to interpret these data, again, this is where it becomes important to communicate with the lab, really look at the data, say these are the data we have from epidemiologists, how do we interpret that root or that common ancestor? Another question is, what is the difference between similar versus related when interpreting a phylogenetic tree? Typically, related means 2 isolates share a common ancestor. So they have a bacterial cell recently at some point in time where they evolved from. Now, related, just like in a human family tree, relationships can be you share the same great-great-great-grandmother or you share the same mother and father. Both of them are related, but related at different time scales. So related refers to common ancestor, while similar simply refers to the number of SNP differences. So you could be related by having the same great-grandparent, but your genetic information could be very similar simply by means of how organisms have become different. Because bacteria and microorganisms that we study here don't follow clear generation times, the interpretation of related versus similar in terms of an epidemiological relationship is not that easy. If you think about families, if you have big enough families that differ enough in variation, You can have relatives that are older or younger, and you can have overlapping generations that can make interpretation very complicated. With the huge differences in generation times you have in bacteria, that becomes an even bigger challenge, and that's why you need to combine epidemiology data with the tree data to really get at identifying outbreaks and identifying sources of outbreaks. Okay. Transition. We got another, I guess, a revised question. Is it possible that an ancestor root defined by generation time could have come after another isolate when defining time in terms of years? Can an unrooted tree be helpful in identifying an outbreak source? So we can build trees, and this gets into the differences of building trees. Trees can be built as unrooted, and it is exactly what it sounds like, or we can root trees, and that's simply a different method of building trees. In terms of identifying a cluster, typically it would not make much differences, but I would probably include an outbreak, an outgroup and root the tree. But in general, because we're really looking at what we call the very ends of the tree, the two of them would not make much of a difference, in my experience. Another one is, I know you mentioned that whole genome MLST and high-quality SNP both correlate well. For some organisms, MLST might work better, and for others, SNP might work better. Is there any value in having both whole genome MLST and a SNP pipeline available for the same organism? My opinion on this one is, as we start out with organisms that we might not have much experience, it will provide some value to initially look at both, understand where they might give us differences, and then what the cause and the root cause of differences is if we see them, and then use that to refine these pipelines. Once we have a relatively mature pipeline, I think it is typically not, or it rarely will be necessary to do both methods. I think at least in the medium time frame, again, if we have large data sets, different labs, we would use whole genome MLST. But if we have, for example, mismatches between epidemiology and our phylogeny and the relatedness derived by these data, then we can run a secondary analysis with a high-quality SNP. So I think we will typically, based on our knowledge, decide which one tends to be the better method, or simply the method that is more rapid, more easy to use, and then use the other method to refine when needed. In many cases, this would be more of a whole genome MLST approach, and some it would be more of a SNP approach, where we then use other methods to refine it. And again, early on, when we don't know much about organisms, there might be some value in running both methods. Okay, with that, appreciate your patience with the technical difficulties we have, and thank you for participating. Again, we will post the webinar. We also post the actual slide set. So if someone wants to download the slide set and use it, it will be available to everyone. Thank you very much.